Welcome, everybody. This is the last webinar for this year, the FARA Flash Talks. I am Liz Sorani. I'm the Director of Research at FARA, and it will be my pleasure today to introduce hopefully 4 speakers. We have 3 connected right now. We'll see if we can get the 4th one to connect. And they will be sharing their research with us. They have 5 minutes and 1 slide to summarize their finding and talk about the focus of their research. After each talk, we'll have a 5 minutes of question and answer, and you can submit your questions or comments by typing into the Q&A box at the bottom of your screen. The webinar is recorded. We will be posted on FARA's YouTube channel, and you can find there our previous 3 webinars as well. At the end of the webinar, you'll be prompted to follow a link to a survey where you'll be asked to rate today's presentation. We also put the link in the chat box. We will recognize the 4 best presentations. So look for announcements from FARA soon to find out which presentation had the most votes. And with that, our co-moderator today is Jeremy Lempicki. Thank you, Jeremy, for helping out today. And would you introduce yourself to our speakers and audience, please?
Sure. Thank you for having me. I'm Jeremy Lempicki, father to Scarlett and Lila, who are both 10 years old and are FA patients, and also team captain for Cousins for a Cure for the upcoming Ride Ataxia in Chicago on July 11th. So a little bit of an advertising plug for Ride Ataxia. They're going to be in person again this year.
Thank you, Jeremy. So let's go ahead and get started. Our first speaker today is Rahman Rafiq. He's at Albany Research Institute with Eric Koppen, and is going to talk about today about cytoskeletal filament proteins in Friedreich's cardiomyopathy. Rahman?
Yes, miss. Can you see the screen?
Yeah.
One moment. Hello, everyone. I am Rahman Rafiq. I'm a postdoc of Albany Research Institute, and I am from Bangladesh. Today, my topic is cytoskeletal filament proteins in Friedreich cardiomyopathy. So we all know that the frataxin deficiency is the main reason for the Friedreich ataxia, and our research question is, where is the frataxin going? Either it is trapping somewhere, or it is transforming, or where it is. So let's dig in. The Friedreich ataxia cardiomyopathy, it's actually cardiac arrhythmias or heart failure, which may occur independently of neurological manifestation of the disease. Our hypothesis of the study, it's frataxin deficiency in heart sets into motion changes in cytoskeletal heart protein, either loss or increase. So in our study, we are focusing on multiple FA-related heart protein, such as Desmond, alpha-bicristallin, beta-spectrin, ankyrin, plectin, myospirin, and JASP. In normal heart, this protein contributes to the mechanochemical properties of the most abundant intermediate heart filament protein, Desmond. And in the for our study, we used the autopsy sample. We preserved in formalin and we also freeze them. The formalin preserved sample, we make the paraffin cassette to conduct the immunohistochemistry study, and the frozen tissue we use to prepare the tissue lysate to conduct Western blots that we are showing the result in figure 1 and figure 2. And the method we use, it's called proteomic, which is systematic analysis of tissue lysate by antibody microarray, Western blot, and immunohistochemistry. Figure 1, double level immunofluorescence confocal microscopy images are showing here. The first row, A is normal heart and B is Friedreich heart. The green are treated with Desmond, red is alpha B crystalline, and the merge is showing on the third one. So the arrow is showing the clear position of intercalated disc, and the fibrils are also evident. But in case of Friedreich heart, the arrow is showing the chaotic disorientation of the intercalated disc, and we also see the fibrosis. In the figure 2, we see the Western blot, our study result. We conducted 2 different study here. The 1, 2, 3 is actually co-immunoprecipitation. That is, we treated the lysates of the Friedreich heart with anti-Desmond and protein A agarose. And we conduct the electrophoresis, and the Western blot, we react with frataxin, that is 1, and 2 and 3, we react with Desmond. And the outcome, we can see that clearly showing in the arrow that there is the band. And we have analyzed these bands with gas chromatography mass spectroscopy, and we found that it is Desmond and Desmond derivatives. So even though we treated with frataxin, we don't see here any frataxin. And 4, 5, 6, we have showing the result that is not co-immunoprecipitation. That is, we use the tissue lysate, and we conducted the electrophoresis and the Western blot. We react with the 4, we react with the frataxin, and the 5 and 6 we react with Desmond, where we can see that frataxin is present. And when we react with Desmond, we see the exact Desmond position at 53, the Desmond band, and other, a Desmond fragment, that is, Desmond has captured other protein here. So our finding of the research is the upregulation of Desmond in Friedreich binds the frataxin, increase the frataxin deficiency. Also, Desmond aggregation contributes to heart failure in Friedreich ataxia. So I would like to take the opportunity to thank my lab, especially my supervisor, Dr. Arnold Koppen. Thanks to FARA, and I am ready to take any question. Thank you.
Thank you for the presentation. What is the sampling method applied here? Is this through a biopsy of the tissue?
We use the autopsy sample,
Okay.
and we preserved in formalin. That is the, when we freeze the sample, it changed the morphology, the physical consistency. So we have to, half of the sample we preserve in formalin, and the half will preserve in minus 80 degrees centigrade, just frozen.
We have a question coming in from the audience from Dr. Sanjay. Have you seen co-localization of frataxin and Desmond in immunohistochemistry?
Yes, that is the co-immunoprecipitation that is showing the result that I have shown in the presentation. We have done like a lot of Western blot study, but here I just present only two result that Desmond is mainly contributing and capturing the other heart related protein.
Okay.
Do you think these changes will also be in muscle? Do you plan to look at, for example, skeletal muscle or other tissues? Do you think this is applicable to other tissues?
This is our we plan to start our the nervous tissue study. Yes, we are going to investigate one by one.
You might have answered this question earlier. Is it just frataxin that's co-localizing with Desmond, or are there other proteins? And do you have ideas as to why it's happening?
Desmond is the predominant in the heart, in heart compared to the other protein. So our goal was like, why is that? Why Desmond is doing it? So that's why we, after finding the result, we convert the gel. We did the Comasi blue staining, and we targeted that band, the particular early band and the later band. Usually Desmond shows the band at 53 kilodalton, but when we do the co-immunoprecipitation, Desmond gives the earlier band, it's above 100, and that band we sent for analysis for the gas chromatography mass spectroscopy, and we figure out, we got the result that it is actually Desmond and Desmond derivatives. Somehow, I am not sure, like Desmond is changing the properties of other protein. This is like maybe just limitation of my understanding, but we are trying to understand more.
I guess I was trying to understand, is this a reactive process, right? Or is the Desmond more primarily contributing to the cardiomyopathy?
That can be. So this is, this is the actual thing, that Desmond is actually, trapping. I am saying trapping, maybe I'm wrong, but somehow Desmond is contributing to, taking other heart protein. And, from our lab, we got the result that Desmond and alpha B crystalline aggregates in the fibrils make it stiffen, which hindered the contraction. We have already published that result. So this is another thing, is like when we checked with other heart protein, and we see that when we do the co-immunoprecipitation, Desmond is actually taking all other protein, somehow.
And do you think that this could lead to investigations on some other potential therapies where we deregulate the amount of Desmond, or would there be some other effects in doing that would create other risks?
Yeah, I strongly believe that, because till now we all know that the deficiency of frataxin is the main reason of Friedreich ataxia. But there are other proteins, playing in this game, and because of lacking of one protein or one gene, the other gets imbalanced. And this is just one finding, that the Desmond is like playing, especially in the heart tissues, in the heart fibrils. So I strongly believe that this is a good finding, and there may be other proteins as well that we have to find it.
Okay. We have one more question from the audience. Desmond is a cytosolic protein that can be found in sarcomere. How do you envision a cytosolic protein co-precipitating a mitochondrial protein?
That will be limitation of my knowledge. Actually, I do not have the answer. I'm sorry, because my background is actually not. I'm not a medical doctor. I'm a toxicologist, and my main basic is like I'm a strong chemistry background. So I'm sorry, I don't have the answer.
It's fine, thank you.
But thank you.
Great, thank you, Ramon. So let's move on to the next speaker, Janetta Matusek. She's a grad student at the Broad Institute with David Liu, and she's going to talk about genome editing, application of genome editing technologies, potential therapeutics of Friedreich's ataxia.
I will share my screen. Can you see it well?
Yeah, perfect.
Okay. Thank you for the introduction. I am Janetta, and I am a graduate student at Harvard University, and I work at the Broad Institute. I am originally from Poland. I've moved to US a few years ago. And today, I would like to tell you about something I'm interested in. I'm interested in genome editing. And the project I will be telling about is related to using genome editing strategies to correct the genetic cause of Friedreich ataxia. So let's look at the genetics of Friedreich ataxia first. Our DNA encodes all the information about the proteins that are going to be produced in our body. Frataxin is one of those proteins, and it's very important for the proper function of mitochondria, which are the powerhouses of the cell. In Friedreich ataxia patients, frataxin protein is downregulated. It's very low, as we just learned. And this low level of frataxin is related then to the Friedreich ataxia-related symptoms. The reason why frataxin is so low is due to the sequence of the DNA coding for frataxin, the frataxin gene. Frataxin gene contains a region which has GAA trinucleotide repeats. Those repeats in Friedreich ataxia patients are very long and can reach up to thousands of units, in comparison to only dozens in case of unaffected individuals. When those repeats are so long, they affect the DNA structure of frataxin gene, which then prevent the frataxin gene to be properly expressed and produce frataxin protein. In case of very, very long GAA repeats, frataxin protein can be not expressed at all. In our project, we are exploring two different genome editing strategies to potentially fix this issue. In strategy A, we are using genome editors to remove the long GAA repetitive tract from frataxin gene in a very precise way, and potentially avoiding unnecessary edits and unnecessary changes to the genome. We expect that those kind of edits and this removal of those repeats will prevent the DNA from forming those atypical structures, and as a consequence, will help increase the expression of frataxin protein. In the second strategy, we are using genome editors to introduce simple changes within the repeats to interrupt the very repetitive character of the sequence. We hypothesize that those kind of changes will prevent DNA, again, from creating those extraordinary structures, and due to that, the level of frataxin protein can be increased. We've done a lot of in vitro experiments to test our strategies and determine editing efficiency in vitro in human cells that encode 30 GAA repeats in frataxin gene. We treat the cells with our reagents, and then we sequence DNA from those samples to determine what actually happened in the genome of those cells. And here I'm showing you the most recent results that we got and we're very excited about. That is showing how well those editing strategies can actually work in this human cell system. So what we saw is that we are able to remove those GAA repetitive tracks from frataxin gene with efficiency of 60%. Additionally, if it comes to introducing those changes within the repetitive sequence, we are able to introduce those edits in up to 50% of all GAA repeats in frataxin gene in human cells. So we've been very excited about this result. And then now the next step would be to test those strategies in vivo in a mouse model of Friedreich ataxia and check what is the editing efficiency in relevant tissues in those animals. And then as well, look at the frataxin expression and then look at the motor function of those treated animals. We hope that if those strategies will show good editing efficiency in mice, we will be able to see increased levels of frataxin protein and then also potential improvement of motor function in those animals. We hope that if successful, those strategies can be potentially used and explored for the therapeutics of Friedreich ataxia in the future. And I'm very excited to be here today. Thank you so much for inviting me to give this flash talk. And now I'm very happy to answer any questions. I'll stop sharing my screen.
Very good. Thank you for the presentation. One thing that you noted, and most of us know, is that patients with FA can have varying degrees of GAA repeats. And if we're editing to remove GAA repeats, does the therapy or treatment need to be customized due to the number of GAA repeats, or does it just seek out any quantity of GAA repeats and attempt to remove those?
Yes, so the strategy that we are taking is hopefully not limited to the number of repeats. So we've been testing it and for this particular number of GAA repeats, but the I guess the assumption is that it should work for any number of those repeats. Of course, for the very long repeats, it might need to be optimized a little bit, but it shouldn't be really a personalized strategy. It should be very universal.
And in a given patient, even on the different alleles, the number of GAA repeats are different. So it would target both alleles and whatever the number of repeats are on each allele.
Exactly, yes.
Okay, very good, thank you. Some questions coming in from the audience. Do the type A and B edits affect cell proliferation, and what cell types were tested?
Yeah, so we have been doing these experiments in HEC cells, HEC293 cells, and also mouse embryonic stem cells so far. And we haven't seen any differences in cell proliferation after those edits, but we weren't also really running any assays that would determine that specifically. It's just when it comes to like our editing procedures, we didn't see any differences between different levels of editing efficiencies and also the behavior of cells in culture.
Okay. Some more questions from the audience. Does the editor B strategy require cells to divide? The target cells for therapy in FA will likely be non-dividing cells.
No, yeah, that's a very good question. Luckily, the strategy B doesn't require the cells to divide. It will be efficient also in cells that are non-dividing cells, like nervous system cells.
Okay. And the next question is, does your excision strategy leave a healthy amount of GAA repeats? If not, is there any known adverse effects to the function of the gene from depleting all GAA repeats?
Yes, that's another good question. So our strategy actually leads to removal of all the repeats, but luckily the sequence that is adjusting to those repeats and that we remove is very short. So we only delete around 30 nucleotides besides those GAA repeats. I am not aware of the literature that would be showing adverse effects of this particular region being removed. Like this whole frataxin gene in that region is very highly regulated and has a lot of regulatory sequences. And I am aware of other genome editing strategies that are targeting that region and are removing way a bigger sequence. I cannot surely say that our strategy is going to not affect the cell metabolism at all, but I think that there is a big chance that it might actually be working well, but we will not know until we test it in animals and in more relevant cells.
Okay. So the next question, I think you probably won't be able to answer until you get to that phase of testing, but do you look at levels of frataxin expression after editing in vitro?
Yes, so unfortunately, the cells that we are testing right now don't have enough of the repeats to actually affect negatively frataxin expression, so we cannot see those differences. We are waiting for iPS cells from Friedreich ataxia patients to test our strategies and then determine if we are able to fix the frataxin level. But yes, I think I will be able to answer surely this question in the next few weeks. But from the literature, we know that the expression of frataxin is actually correlated, like inversely correlated with the number of repeats. And we hope that removal of that region, that it was also shown in other literature, can help to increase the frataxin. So if we believe in this, like when we consider this assumption and we translate our strategies, I think we could expect actually, I mean, we expect to see increased frataxin expression, but we need to test it, of course.
Okay. And you touched on briefly about some of the risks or concerns about making unintended edits, and what are the strategies there? How do you avoid that?
Yeah, so the first strategy I was telling about is actually very precise in introducing those edits. It's the level of indels, which are the side effects of genome editing, is very low. And we have a high control over what editing outcomes we can get from this particular editing strategy. In terms of strategy B, we are avoiding cutting DNA at all. So in strategy B, we are actually minimizing any unintended edits, indels to the genome whatsoever. So hopefully that will be a good thing.
And with strategy B, you described it as like creating an interruption, correct? And
yes.
Is that interruption all along all of the GAE repeat sequence, or is it just in certain areas?
So our strategy is targeting the whole length of the GAE repeats. We can see that actual edits are focused in particular spots, in particular regions of the GAE repetitive tract. And we can also see a correlation of the editing efficiency and like how many repeats are actually getting edited with the length of the repetitive tract. But it's never like very, very dense. So we are hoping that since this strategy is interrupting the repeats across the board, but also is not super intense with doing so, we hope that it will not be enough of those interruptions to actually create any problems.
Okay, thank you.
Thank you.
Thank you, Janeta. That was a nice discussion. So I see Karen joined us. So the next speaker is Karen Girotto. She's at University of Campinas. Actually, I'm not sure. She used to be a previous, she was a grad student with Marcondes Franza, but I'm not sure where she is right now. But she's going to talk about work she's done with Dr. Franza on preclinical left ventricular mitochondria remodeling in patients with Friedreich's ataxia, a cardiac MRI study.
Well, hello, I am Karen. I need to share my screen. Just a minute. Yes.
Yes, there it is.
Yes, there it is. Okay. My name is Karen, and I am a neurologist here in Brazil. I work with Professor Marcos França, and I'd like to thank you for this opportunity. I'm here to talk to you about my work entitled Preclinical Left Ventricular Myocardial Remodeling in Patients with Friedreich's Ataxia, a cardiac MRI study. So we know that cardiac involvement is frequent in FRDA patients, and heart failure is considered the leading cause of death in this population. We know that concentric increase of ventricular wall thickness is related to cardiomyopathy and leading to heart failure, which is a major problem in these patients. We know that proliferation of myocardial fibrosis, which is a scar in the heart tissue, is a hallmark of FRD's cardiomyopathy with clinical heart failure. And we know that cardiac magnetic resonance, or CMR, is a promising diagnostic technique. And this technique helps us to assess left ventricular remodeling and also expansion of fibrosis. Through this technique, we call it ECV, or extracellular volume fraction, and CMR can help us study cardiomyocyte size. Through this technique, we call it tau. So in this work, to do that, we recruited 27 patients, FRDA patients. Mean age of these patients was 27 years old, and 15 of them were female, and they did not have heart failure. And we also recruited 10 health volunteers with mean age 32 years. 5 of them were women. We proceeded with clinical and neurological examination. We assessed FARA scale, and then they proceeded with CMR, including assessment of myocardial extracellular volume fraction. And we studied intracellular lifetime of water, tau, which is a measure of cardiomyocyte size. We also classified cardiomyopathy using Weidmann's classification. And we have found very interesting results. So we have seen that FRDA patients had normal left ventricular ejection fraction. So this means that the heart of these patients could pump blood normally, but they have significant increased left ventricular mass index. We have studied ECV, which is a measure related to fibrosis, and cardiomyocyte size. We use it tau technique. And we have seen that these measures were larger in FRDA patients when we compare with controls. Also, we have seen, like we see in the figure, that LV mass index to and the diastolic volume ratio in FRDA patients was significantly higher than in controls. And this is this indicates a hypertrophic phenotype. Also, we have seen that patients with late onset FRDA had higher tau values. So they had cardiomyocyte, the size of the cardiomyocyte was higher in late onset patients than in patients with normal beginning of the disease. And we have also seen that ECV, or the extracellular volume, correlates with Weidmann's classification. So we have seen that fibrosis and also hypertrophy was very important cause, is importantly related to cardiomyopathy, hypertrophic cardiomyopathy in these patients. When we think that late onset FRDA patients had higher tau values, we kept thinking that maybe patients with shorter disease duration show this higher tau volume. So we think maybe FRD cardiomyopathy may present with cardiomyocyte hypertrophy, and in a later phase, it manifests itself by a regression of cardiomyocyte hypertrophy or possibly LV atrophy and LV dilation. So this could potentially explain why patients diagnosed earlier and with longer disease duration have a lower tau. We know that in mouse models of FRDA, myofibrils are atrophied, and the initial cardiac hypertrophy has developed into a dilated cardiomyopathy at the onset of cardiac heart failure. So we concluded that extracellular volume fraction, which is a measure of fibrosis, and tau, which is a measure of the size of the cardiomyocyte, is associated with more severe cardiomyopathy classification, suggesting a role of these markers in tracking disease progression. Thank you very much. I was a little nervous during the presentation, but I thank you very much for the opportunity, not only to present my work, but also to work with FRDA patients. It's been a great pleasure for me. Thank you.
Thank you for your presentation. So I think what you've stated here is you've evaluated patients just at one point in time, but you haven't done a study yet, say at a 6-month, 12-month interval to measure change in any of these measures, correct?
Sorry, can you repeat, please? I couldn't hear properly.
So from what I understand is that these measures that you've taken were just taken once with these patients, and you haven't monitored these patients over a period of time, say at a 6-month or 12-month follow-up to see if there were any changes in any of these values, correct?
Yes, in fact, I have evaluated these patients only once, but my Professor Marco Speranza is continuing this study, and he's evaluating them again, but the results are not ready yet.
Okay, which kind of leads into some of the questions that we're receiving from the audience. So is ECV progressive, and could it be used as a biomarker for therapeutic response?
We believe so, that ECV is a very interesting technique we're using in CMR, and we believe it can be a useful biomarker for FRDA. Tau is also very, very interesting. It is actually, we have a notion of the size of the myocyte through this technique, which call it tau, and I believe both are very promising biomarkers.
Okay. And you mentioned that you were, say, monitoring data as it relates to age of onset, but were you also looking at number of GA repeats and how there might be a correlation there? And we typically know that longer GA repeats generally lead to an earlier age of onset, but is that something that you also documented as part of your studies as well?
In our patients, there was no correlation between ECV or tau and GAA. We have studied that, but there was no correlation.
Okay. And then another question that we have from the audience is, do you see a gender difference in cardiac function in FA patients?
No, we have found no difference.
Okay. I think the last question we kind of already touched on, but again, asking if these markers could be good for tracking disease progression, and could they be used as endpoints to evaluate potential therapies, which I think you stated in your previous answer that, yes, you do believe that these have the potential to offer that for, being a biomarker or endpoint for future clinical trials.
Yes, we believe so. We believe it's a very promising technique.
Okay, very good. Thank you.
Thank you very much.
Karen, just one quick question, and if you can maybe give us speculate on what do you think the sizes are big at the beginning, the sizes of the cardiomyocyte, and then they decrease. What do you think happens? Is this some kind of compensatory mechanism that then goes away? Do you know? You have any clue?
We believe that maybe, how do I say, maybe extenuate, I don't know how, it's only a hypothesis. We don't have, we still need to study more this subject.
Okay, thank you.
Thank you.
So our last speaker is Jamison Seabury. She's at the University of Rochester. She's a clinical research coordinator with Chad Eatwell. And she's going to talk about new instruments, the Friedreich's Ataxia health indices, a novel outcome for measuring how a patient feels and functions in clinical trials.
Thank you. Can you see my screen okay?
Yeah.
Great. All right. My name is Jamison Seabury, and I'm a research coordinator with Dr. Chad Heatwell's Clinical Outcomes Lab at the University of Rochester. I'm pleased to present to you our FARA-funded research, the development of the Friedreich's Ataxia health indices, which are novel outcome measures designed to monitor how an FA patient functions and feels during a clinical trial. Our team's work is centered on developing disease-specific outcome measures for neuromuscular disease populations. We've now developed over 100 disease-specific outcome measures, which have been licensed for use in clinical trials worldwide by academic partners, industry, government agencies, and foundations. So why is this work important? Well, disease-specific outcome measures allow patients and caregivers to provide a voice into therapeutic benefit. They have the potential to reduce study sample size requirements. They're highly responsive to clinically relevant changes in health, and they can encourage pharmaceutical interest in FA therapeutic development. As therapies for FA are developed, it's really important that researchers have outcome measures that can reliably detect patient-meaningful changes in FA disease burden. Our research is currently developing and validating 2 outcome measures for FA, one for patients ages 11 and older, and one for caregivers of pediatric FA patients ages 5 to 21. We're using the same methodology that we have used for the many other disease populations to create and validate the FA health indices. An overview of this process is shown on the left in Figure 1. We began this work by conducting qualitative interviews with 15 adults with FA, 14 minors with FA, and 10 caregivers of pediatric FA patients. In these interviews, we asked about which symptoms of FA are most impactful on an FA patient's quality of life and disease burden. We asked questions about the physical, social, emotional, and disease-specific areas of patient health. From these interviews, we obtained over 2,500 quotes regarding the symptoms that are most important to the FA population. We then conducted 2 survey studies where we asked FA patients ages 11 and up and caregivers of pediatric FA patients to complete a survey which asked about the symptoms identified from the qualitative interviews. For each symptom question on the survey, patients were asked, how much does the following impact your life now? And for caregivers, how much does the following impact your child's life now? 153 patients and 49 caregivers from FARA's registry completed our surveys. From these responses, we assessed the average prevalence and mean impact of each symptom and symptomatic theme among a larger FA patient and caregiver population. Figures 2 and 3 show symptomatic theme prevalence and impact as reported by patients and caregivers. Using this cross-sectional study data, we developed the first versions of the FA patient-reported health index and the caregiver-reported health index. The FA patient health index consists of 114 symptom questions that measure 18 distinct areas of FA health as well as overall disease burden. The FA Caregiver Health Index consists of 92 symptom questions that measure 19 distinct areas of FA patient health and overall disease burden. The FA Caregiver instrument measures all 18 of the same subscales as the patient instrument, plus one additional subscale, which is attention. Next, we conducted interviews with 17 FA patients to assess comprehension and usability of the FA patient instrument. Participants were asked to provide feedback and comment on the ability of the instrument to measure how they feel and function. We used the feedback we received from patients to make minor modifications to the instrument. 6 questions were deleted, 5 questions were added, and 2 questions were reworded. Overall, patients felt that the FA High was able to capture how they function and how they feel. Patients also commented that they found the instrument to be clear, comprehensive, easy to use, and highly relevant to them. Retest reliability testing of the FA High patient instrument is currently underway. We will also conduct beta interviews with FA caregivers to assess comprehension and usability of the caregiver instrument. We expect that both the patient and the caregiver instruments will be finalized by the end of the year. Overall, we anticipate that the final FA health indices will be highly responsive, relevant, patient-centered mechanisms to measure clinically relevant changes over time. This research would not be possible without the funding and support from FARA. We'd also like to acknowledge the participation of hundreds of FARA patients and caregivers who dedicated their time to this project. We're extremely grateful for your time and support. Please reach out to us with any questions or comments. Our contact information is at the bottom left of the poster. Thank you.
Thank you for the presentation, and I think it's important to continue to focus on efforts such as this. Seeing the power of the voice of the patient and our efforts to move along. Some of the research with RIATA and FDA, it can make a difference, and we can always talk about and measure biochemical changes in patients, but understanding the outcome and how they're feeling, what benefits they're gaining from it, are equally as important to help those studies move along. Do you see this as something that can, be supplemental to the MFARS and should be considered as something that's monitored during the natural history studies that are ongoing?
Yes, absolutely. We expect that this will supplement existing infrastructure that's out there in the FA research community. This was developed by patients and for patients to track small but clinically relevant changes over time, and we hope to facilitate widespread use of the instruments upon completion.
Okay. And then we have a question from the audience. Did the caregiver and patient reports correlate in assessing health indices?
So the patient and caregiver, the surveys were developed independently. So these were not matched interviews. Further research could be needed to match patient and caregiver responses, but these were developed as independent outcome measures.
Okay. And then a question came up as well that it sounds as though the survey was created in English. Is there plans to make this available in other languages and regions via translation?
Yes, so for previous instruments that we've developed, we began by developing the instrument in English and then facilitating translations and cultural adaptations in other languages. And we currently have several outcome measures that are translated in over 10 different languages.
Okay. And then how do you manage whether the individual taking the survey may just be having a good day versus a bad day? That's always a challenge with even some of the questions related to MFARS. How do you factor that in and how do you take that into account?
Yeah, absolutely. So these questions are selected to specifically to track therapeutic benefit. And one of our validation steps is to conduct retest reliability. So we administer the survey twice over 2 weeks. And so questions that fluctuate significantly over that period of time are not chosen for inclusion in the instrument.
And how much time do you think you would need to pass between surveys to be able to observe or measure some meaningful change in the survey? Obviously, if you take the survey and the span of just 1 month, you probably won't see a lot of change. But what rate of time lapse between surveys do you think you need to see changes?
So we have plans to, our next objective will be to observe how the instrument performs over time. So a longitudinal study will be needed to determine that the sensitivity of the questions over time, and what time points do we see a significant change.
Okay. And then recently, a FA app was rolled out for the community to download and use. Any thoughts or discussions about how to possibly integrate it into that and collect data from a broader audience?
Absolutely. I think making these surveys available remotely, that's something that we've done already with our research, and especially in the COVID world, increasing access to these outcome measures, perhaps in the context of a clinical trial, that'll be really important as we move forward. And so that's definitely something that we'll consider for implementation.
Okay, thank you.
So Jameson, were there symptoms that were not in the initial survey but turned out to be important for patients that you added afterwards?
So there were a few questions during our beta interviews that we got some recurring feedback that patients felt that they were important. So they were added back from our original survey.
Great. Thank you so much.
Thank you.
Okay, so we came to the end. So let me thank the speakers again for sharing their research, and thank you so much, Jeremy, for moderating with us. I know a lot of you share this thought with me, and I would like to say that I am so impressed with the talent and the dedication of the young scientists who have presented in the last 4 weeks. And we do hope that they'll continue to work on FA during their careers. Thank you, all of you who attended. Thank you for celebrating FA Awareness Month with us. Remember to fill out the survey, and we will see you next May.
Yeah, thank you to everyone.
Thank you very much.